Good morning, good afternoon, good evening. Today we are going to discuss next unit, that is your with respect to blood. So in blood, what is the composition of blood? Blood contains the cells, and cells in the bloods are RBCs, WBCs and platelets. RBCs we also called erythrocytes. WBC we also called leukocytes, and platelets we also called thrombocytes. Sites indicate it's a cell. Site, जिन words के पीछे site लगा हो, that are the cells, whatever it is, right? So we call it erythrocytes, leukocytes or thrombocytes, right? So these are the cells which are there in the blood. There is a plasma as well, and plasma contains proteins as well, right? So one by one we will discuss in details RBCs, then WBCs, then platelets, and what are their role. So if you talk about RBCs, the role and functions of the RBCs. So RBCs provides the oxygens and nutrients to the cells, right? So how it provide? There is a cardiac cycle, many times we have discussed. This is your left atrium, this is your left ventricle, this is your right atrium, right ventricle, right? So this left ventricle will push the blood to the aorta to arteries, to capillaries, and through capillaries there is a exchange in the, for example, this is your group of cells. So your blood will exchange the oxygens and nutrients. So oxygen will be taken by the cells, and that oxygen will be used for the oxidative phosphorylation, Krebs cycles, and nutrients will be utilized. And in response to that, these cells will release some, like carbon dioxide, it will release waste products. So this is your oxygenated blood which your left ventricle is pushing, and oxygenated blood means it's a 95% oxygen and 5% is your carbon dioxide, right? So when this exchange happens, now this blood contains the deoxygenated blood, means 95% your carbon dioxide and 5% your oxygen. And this deoxygenated blood will go to the right atrium, then right ventricles, and right ventricles it will goes to the lungs, right? Lungs, and then enter into the left ventricle. So in the lungs, this carbon dioxide will be exhaled and oxygen will be inhaled. So oxygen, this is a cardiac cycle, right? कि आप ऑक्सीजन ले रहे हो, कार्बन डाइऑक्साइड छोड़ रहे हो, तो लंग्स में आपका दिस deoxygenated blood will convert into the oxygenated blood, then oxygenated blood will enter into the left atrium से left ventricle, then ventricles will contract and push the blood to the aorta, arteries, capillaries. In capillaries there is exchange of oxygen and nutrients. In response to that, carbon dioxide will be released and waste products, and that blood is deoxygenated, and this is your cycle, right? So this RBCs is contains the hemoglobins that bind oxygen molecules, so oxygen transport will happen. Nutrients are also transporting, right? So this is your function of your RBCs. So but with respect to RBCs, if you go for a laboratory test, so in laboratories you will find various parameters with respect to RBCs has been done. One is your PCV. PCV is your packed cell volume. Or we also called it hematocrit. So if you took a blood in a capillaries, right, not a capillary, in a tube, right, in a anticoagulant tube, the tube which contains the anticoagulant. So you took the blood in the tube, and this tube contains the anticoagulant. Otherwise if you are putting in a simple tube, your blood is going to coagulate. That is the intrinsic property of your blood. So if you are taking the blood in a tube which contains the anticoagulant, so what will happen? Your blood will not clot, and you have done the centrifugations. You have centrifuged the tube. So after centrifugations, you will find that cell will be settled down. RBCs will be settled down, and above RBCs you will get one buffy coat, that is your WBCs, and above that you will get the plasma after centrifugations. So you took the blood in a tube उसके बाद आपने उस ब्लड को सेंट्रीफ्यूज कर दिया, सेंट्रीफ्यूज करने के बाद जो सेल्स होंगे वो सेटल डाउन हो जाएंगे आरबीसी नीचे, ऊपर बफी कोट होगा दैट आर योर डब्ल्यूबीसी, और ऊपर आपका प्लाज्मा आ जाएगा, राइट। सो व्हाट इज पैक्ड सेल वॉल्यूम? सो पैक्ड सेल वॉल्यूम एज इट्स नेम इंडिकेट, पैक्ड और किसका वॉल्यूम है? सेल्स। So the volume which is occupied by your RBCs in a packed condition, that is we call packed cell volume, or also we call it a hematocrit, right? So packed, if you took for example 1 ml blood and you have centrifuged the 1 ml blood, so packed cell volume, the volume, this is the volume which is occupied by your cells, which is occupied by your RBCs, right? So in 1 ml blood the packed cell volume is 0.45 in a healthy, 0.45 ml. This is the packed cell volume, right? So in 0.45 ml is your packed cell volume if you are if you are doing the centrifugations of 1 ml blood. If you are taking 10 ml, then it will become 4.5 ml. If you are taking 100 ml blood, then it will become 45 ml. That is your packed cell volume, right? Volume which is occupied by your RBCs in a packed condition. So how it will be packed? You have to do the centrifugations, your cell will settle down, it will be packed, and normally it will be 0.45, right, in 1 ml blood. So that is your packed cell volume. So I hope you understand what is packed cell volume, or we also called it hematocrit. Hematocrit. So now for examples, there is a two patients, patients number 1, that is anemic patients. Anemic means their RBC is decreased, right? So if you are going to measure their packed cell volume, so for example, of course these are your anemic patients, so your packed cell volume will be decreased. So for example, you are getting 0.25, right? It's less than 0.45, right? So in another one also you are getting 0.25, right? Got it? So 0.45, 0.25. So it indicates that the patient is anemic because their volume is decreased. So now when you do the packed cell volume, so for example in these patients the RBC size is increased. So RBC size is increased, and in this case RBC cell size is decreased. So you know, in same volume, these patients in same volume is same, it can have a less number of RBCs, right? Because your RBCs cell size is more. वॉल्यूम आपका है 0.25, उस वॉल्यूम में अगर आपके सेल्स का साइज बड़ा है तो कम सेल्स आएंगे उसमें, अगर आपके सेल्स का साइज छोटा है तो सेम वॉल्यूम में आपके सेल्स ज़्यादा अकोमोडेट होंगे, राइट? सो दैट्स व्हाई आफ्टर दिस हेमेटोक्रेट you have to see the RBC count as well, RBC count of the patients as well. So why RBC count is important? क्योंकि हो सकता है दोनों patients का packed cell volume same है, but their number can be different. RBC number can be different because for example this patient is macrocytic anemic patients, this is microcytic. Micro means छोटा size के RBCs हो गए, इसमें बड़े size के, तो वॉल्यूम आपका सेम है पर सेम वॉल्यूम में अगर साइज बड़ा रहेगा तो कम सेल्स आएंगे, अगर साइज छोटा रहा तो ज़्यादा सेल्स आएंगे. सो दैट्स व्हाई आरबीसी काउंट इज ऑल्सो इंपॉर्टेंट. सो यू हैव टू डू द आरबीसी काउंट एज वेल. By I think you are doing in your human anatomy and physiology lab by hemocytometers, RBC count, WBC count, right? Then there is a third thing is you have to measure the hemoglobin concentrations. Hemoglobin. So hemoglobins can be different even though if your packed cell volume is same, your for example number of RBC is also same. For these are the two patients, their packed cell volume is same, their RBC's number is also same, but they can differ in their hemoglobin concentrations. So hemoglobin is a component which is present inside the RBCs. So maybe this patient's hemoglobin is less, this patient's hemoglobin is little bit more as compared to this. So patients can be different in their hemoglobin concentration even though their packed cell volume and number of cells can be same, right? So hemoglobin is a component which is of one third of your packed cell volume. So hemoglobin is loaded in your RBCs 1/3 of your packed cell volume. So if you are taking 1 ml blood, as I said, in normal individual, so packed cell volume is how much? 0.45. So 1/3 of this is 0.15. So 0.15 gram of the hemoglobin in 1 ml blood. And if you are doing 10 ml, then it's 1.5. If you are taking 100 ml blood, so in 100 ml blood, how much hemoglobin? There is a 15 grams. Because your packed cell volume is how much? 45. 45 का 1/3 कितना हुआ? 15. 15 3 45, right? So 15 gram in 100 ml blood. So accordingly you can do the calculations, right? So this is hemoglobin. So now there are some RBCs indices which you are deriving from these values, like MCV, MCH. This is the mean corpuscular volume. This is the mean corpuscular hemoglobin. So how you can calculate? You can calculate if you know the number of RBC, if you know the PCV values. So if you know the PCV value, this is mean corpuscular volume is what? It is the volume of the RBCs which is occupied by the single RBCs in the packed situation. Single RBC. So how you can calculate? So MCV is equal to PCV, packed cell volume, divided by number of RBCs. So RBCs are in billions, you know. So of course your numbers will be very less. Like MCH is equal to hemoglobin divided by number of RBCs. So these are the indices which you can derive if you know the hemoglobin value, if you know the packed cell volume. So you can calculate these MCH, MCV, right? So that is your mean corpuscular volume and that is your mean corpuscular hemoglobin. So this is about your RBCs and RBC indices. Next video we will discuss about WBCs and what is the role of WBCs in the immunities and what is their mechanism. Thank you.